why cells are the size that they are and why that matters, and as an activity to investigate the effect of cell size and diffusion. So diffusion is the passage of particles from an area of high concentration to one of lower. And with a cell, this is how they get nutrients in and waste materials out. So cells need to have a large surface area to provide for all the needs of the cell. But as the cell will get larger, the cytoplasm inside the cell, furthest from the cell membrane, might receive nutrients too slowly to perform its metabolic functions. So the volume of a cell determines the requirements, the needs of that cell, and the surface area determines the ability to supply it. So we're going to look at the surface area to volume ratio and how that affects the size of the cell. So the rate of exchange of materials, and indeed energy, is a function of its surface area. And so for a sphere, not that all cells are spherical, we're going to look at the surface area of the cell, of the sphere, 4 pi r squared, and the volume 4/3 pi r cubed. And so if we put surface area over volume and put those formulas together, we end up with 3 over r for the surface area to volume ratio. So 3 divided by the radius. Now pause and think what's going to happen to the ratio as the radius r increases, what effect that would have on cell size. And so nutrients are entering the spherical cell by diffusion, and what happens to nutrient uptakes in larger cells. Remember the surface area SA over V ratio is 3 over R. As the denominator R gets larger, the ratio is actually going to get smaller. So is there a limit to the amount of nutrients that can be taken into a cell just investigating surface area to volume ratios? So the one on the left, radius r, formula for surface area and volume, and the surface area to volume ratio is 3 over r. Now when the radius, can we say r equals 2 times the radius? So we're putting 2r into the formulas instead of just r. What happens to that middle cell? So try and work that out in terms of algebra to work out what happens to the surface to volume ratio. And pause while you do that. So our surface area is now 4 pi times 2r all squared, which is 4r squared. And our volume is 4/3 pi 2r all cubed, which is 8r cubed. And so when you put them and cancel out, you get 3 over 2r. And finally, for 3 times, pause while you work out, and you'll put 3r, whereas in the first one you just had a single r. So here we've got 4 pi 3r all squared, that's 9r squared for the surface area. And you've got 4/3 pi 3r all cubed, which is going to be 27r cubed. And when you do that calculation, you end up with 1 over r. And the volume is 27 times that of the single r radius, comparing it. So we're going to simulate diffusion in cells of different sizes with gelatin cubes. made with the addition of an indicator. And the acid-base indicator will change colour in the presence of an acid such as vinegar, acetic acid. So the surface is going to change colour, and in time the acid reaction with the indicator diffuses through the cube, and more of the cube changes colour from the outside inwards. And you'll use the data that you collect to determine the optimum cell size, gelatin cube size here, in the time allocated. And so let's look at a cube. We've looked at spheres. It's a cube, so all the sides are the same, and for the surface area there are 6 squares, 6 times L squared, and the cube volume is L times L times L, L cubed. And so what is the surface area to volume ratio when L equals 1? So pause while you do that. And what happens to the surface area to volume ratio as the length L increases, doubles or triples? Again, pause to work that one out. So in our lab, why are the cells not the size of elephants, or elephant just be having a single cell? You're going to choose 5 sizes of cube. You will cut them to the sides you determine, and use metric measurements. And each type, different cube size, will have a different surface area and volume ratio. One must be a 1 centimetre cube, and don't go larger than 2.5 centimetre sides on the cube. 3 trials for each, and cut out your cubes. So materials, safety materials, gloves, goggles, aprons, and a slab of gel made with gelatin and an acid-base indicator. A mounted blade to be handled carefully and put down. If you've got a dissection tray, place it at one side of that, out of where you're going to be handling the cubes. Beakers and some white vinegar, 5% acetic acid, and a 15 centimetre ruler. And here are a collection of the cubes that you cut. Cut them as carefully and accurately as you can. For safety, wear gloves and goggles, and care when handling the mounted razor, and keep your goggles on even through cleanup. Procedure: cut the cubes, place them in a beaker, add enough to go into the to cover the cube, and then use the same volume in all of your beakers. After 10 minutes, and you can do gentle stirring of the solution around the cubes, use tongs gently to remove the cubes from the acetic acid, and you can rinse them and blot them with paper toweling. Don't squash or damage the cubes as you do that. So you're going to cut each in half and measure the width of the inner blue cube, which hasn't changed colour, hasn't been penetrated by the vinegar, and you already know the actual size of the cube that you started with, and record the resulting volumes in the data table. So here we are measuring the vinegar to put into each of the beakers. You can look at the measurements on the beaker to give you an indicator of how much you need to add. So there are your acetic acid vinegar solutions ready to go, and with each of the cubes inside. Time when you put the gelatin blocks into the acid, and you may gently stir, and then you have to take them out, blot them, and measure them. And you can rinse them with distilled water, and then cut each block in half, record the whole diameter, which you should know from your original cutting measurements, and then the unpenetrated width, here the blue where it hasn't changed colour. Data table, and you can pause this to read the calculation assist below so that you know what you're putting in each of the columns. And the cube length will be one of your 5 different cube widths. So, what would the gelatin cube represent in an organism, and what does the vinegar represent? And in terms of maximizing diffusion, for the results you got, what was the most effective size cube that you tested? And write down the data as you answer that. So which cells would be able to survive and which cells would die, and state how you drew this conclusion. And a little bit more about cube calculations. We've done the surface area of 1 cube, and surface area to volume is 6:1. And with 8 1 centimetre cubes, so the length of each side is 2 centimetres, work out the surface area to volume ratio, that middle set of blocks, and pause while you do that. And then go on to the third, where you've got 27 1 centimetre blocks sacked into a cube. Each side is 3 centimetres long. Work out the surface area to volume ratio, and you come up with 2:1. So for 1 centimetre, 6:1, 2 centimetres, 3:1, and 3 centimetre cubed, 2:1. And using this same method, work out the surface area to volume ratio for a solid cube made with 4 1 centimetre blocks along the side of the face, of each face. So what cell shape, cube or sphere, is worse for keeping a cell alive? Which is the best? And again, explain your reasoning. So why do organisms grow by increasing the number of cells? Why not simply increase the size of their existing cells? So diffusion works well in smaller unicellular units. Diffusion isn't sufficiently effective to get nutrients in and excretory materials out in a manageable time. So if organisms are much larger, specialized organs assist with nutrition and excretion. And all living cells are surrounded by aqueous solutions, and the cells contain different concentrations of solutions, the cytoplasm. So with increasing cell size, the radius, less of the cell's interior, its cytoplasm, has access to the cell's surface area for, we'll say, rapid enough gas exchange, waste material excretion. And we're not looking at single cells here for everything, but just relative sizes of organisms, and the scale is a logarithmic one. So each one is 10 times the one before that, and 100 times the one two before that, etc. And at the bottom, how you can see these. It's pretty tricky to see an atom with an electron microscope, but a scanning tunneling microscope, you can get some measure of it. And with Very many thanks to Washington International School, Miss Shaw's upper school biology class. Thank you.